Good morning, everyone. I am delighted to have the honor of introducing April Stempian-Otero this morning. April began her career as an East Coaster, graduating from Dartmouth, unsurprisingly given the title of her talk, with a degree in English literature. She did her medical training at the University of Connecticut and the University of Virginia. And fortunately for us, she was wooed out to Seattle for cardiology fellowship, where we've been lucky to keep her since. I have known April for nearly 20 years. And like for so many others in this audience, she was one of my interviewers. And it was really a lot of her energy, humor, and how inspirational and passionate she is about the promise of academic medicine that was a good part of why I and many others came to UW. One of her many less heralded accomplishments has been her commitment to mentorship. And this has been shown not only in her own translational research lab, her work as APD for research in cardiology and as a PhD advisor, but also in things like placing URM high school students in science labs for the summer. This is also shown more informally and yet no less importantly, over and over again, in how she uses both her enormous network and her genius for knowing who might work well with whom to connect people and make all of our work better. She is truly one of the most generous people I've ever known. With respect to our own research, the underlying developmental mechanisms of fibrosis and heart failure and the contribution of inflammatory mediators has been April's predominant focus ever since even her earliest KO8 just after fellowship. Her work has been exceptionally productive in moving forward our understanding of these complex relationships. And we're really fortunate to be able to learn from her today about little fires everywhere, immune activation and chronic heart failure. I will remind you that you can put questions in the chat, and when it's time to ask them, I will be able to unmute you. Thank you so much for being here today.
Chris, that was so generous, I may cry. I have to get rid of the looking at me. Okay. I'm so sad that we don't have grand rounds in person because I really am somebody who gets energy from my audience. And when I'm talking about complex concepts, I like to look out and see people that are not PhDs and not MDs nodding and smiling. And I know that I have hit the right bar of explaining what I want to explain. So if some of you could nod and smile and just send that to me, so I know you're out there, even if I can't see you, I'd appreciate it. So yes, immune activation and chronic heart failure. So let's see, here we go. So I'm starting with the immune system, and I was really taken this summer. The Atlantic had this article on COVID, and I do have to say that the COVID pandemic, for its good and its bad, has really revitalized my interest in studying inflammation in the heart. So the Atlantic had an article, Immunology is where intuition goes to die. And the first paragraph is this joke. There's a joke in immunology, which Jessica Metcalf of Princeton told me. An immunologist and a cardiologist are kidnapped. The kidnappers threaten to shoot one of them, but promise to spare whoever's made the greater contribution to humanity. The cardiologist says, well, I've identified drugs that have saved the millions of people. I think he's talking about thrombolytics. And look at that, I'm assuming it's a he. Impressed, the kidnappers turn to the immunologist. What have you done, they ask. The immunologist says, the thing is, the immune system is very complicated. And the cardiologist says, just shoot me now. So hopefully nobody is going into this talk thinking that I'm going to talk about immunology and it's the most complicated, obtuse thing in the world, because I'm going to hopefully get you excited about it. So the goals that I'm going to talk about, innate versus adaptive immunity, the role of innate immunity and macrophages in homeostasis in the heart, the response to injury, the chronic heart failure issue, starting with the TNF data, and some unanswered questions. So I like to think, I came up with this analogy about innate versus adaptive immunity. And I'll say that first, when I put these pictures up, I was like, oh my God, that's so sexist, this macho man and this, delicate woman. But every single analogy that's out there about the immune system is all war. It's, you've got patrol people versus Navy SEALs coming in. And so I'm going to stick with this one because it's not war and it's not police and it's not violent. So the innate immune system is the immediate picker-upper in your body. It cleans and disinfects things that spill on the floor. The adaptive immune system I look at as tidying up with Marie Kondo. It organizes what stays and what goes. It looks at things around and says, should this stay? Should this go? So at this point, if you're not interested in any more details, you can turn off for a while till you see these pictures again. So again, innate immunity, a very ancient program. From the time organisms became multicellular, macrophage monocyte-like cells developed and an innate immune system was there. The adaptive immune system does not appear in evolution until jawed vertebrates. Key thing, there are vertebrates without jaws, until jawed vertebrates. So it developed quite a bit later in evolution. The innate immune system, as I implied, its goal is the rapid detection and containment of tissue injury and infection, i.e. the first line of defense, very quick and very simple-ish. Adaptive immune system is the antigen-specific response. It takes longer to develop, and it has multi-pronged strategies to clear an infection or clear up damage. The innate immune system's goal is to clean up the damage and transition to tissue repair and regeneration. That's really its main goal, is cleaning up things, making sure they're not infected, regenerating the damaged tissue. Whereas the adaptive immune system has a major side goal of generating memory to that injury, that virus, that bacteria, to accelerate the response to the next insult. In innate immunity, the primary cell is the tissue macrophage. In adaptive immunity, the primary cell are the lymphocytes. In innate immunity, the triggers to this system are a variety of proteins that are generally associated with infection or injury. So they're called patterns, pattern receptors, in which it's a pattern of a protein that's triggered the system, as opposed to the adaptive immune system, where very specific pieces of protein, sequences called epitopes, are what triggers the adaptive immune system. The innate immune system has no ability to detect self versus non-self because of this. It just looks at these patterns of proteins, whereas the adaptive has the major value of being able to differentiate self versus non-self. Now, as a transplant cardiologist, I spent a lot of time with the adaptive immune system and trying to hide the non-self heart from the self adaptive immune system, but the two are interrelated. So schematically, here's innate immunity. And this is skin, because you can see that the tissue macrophage in this case is a Langerhans cell in the skin. So you have an injury, presumably with many organs. This injury is accompanied by bacteria coming in. Like if it's skin, there's bacteria, lungs, there's bacteria, mouth, lots of bacteria. But you have a combination of injury. The injury induces these these cells to become activated. They secrete cytokines out into the world. The cytokines then stimulate circulating granulocytes and monocytes to traffic in to clean up the injury. The adaptive immune system is quite a bit more complicated. And it feels like there's cells being discovered on every other year basis. As you can see here, we're down to many types of CD4 cells at this point in time. I think there's 6 different CD4 cells. I'm continually taken by the fact that we went from TH1, 2, and 3 to 17, and I don't know what happened to 4 through 16. They were named something else, I guess. But it's a very complex system, and as you can see, it's multi-pronged. It can kill cells through cytotoxicity, or cells or foreign materials, as plasma cells at one end to make immunoglobulins, to also attack foreigners, and is set up by these dendritic cells that present the antigen to start the system. Now, these two overlap, of course, as I mentioned, that the dendritic cell, which is stimulated by innate immune system, takes in the damaged pieces or virus or bacteria, traffics then to a lymph node, where in the lymph node it presents this epitope, this specific protein, to the T cell immune cell. And it presents it in the context of MHC, self or non-self. So at this point, the T cell recognizes this is non-self and is going to stimulate these different pathways to both regulate the immune attack in the moment and also to create memory for the future attacks of this immune system. Both of these have negative feedback mechanisms because obviously if they go wild, it's not good. And we do have many diseases where dysregulation occurs. So in innate immunity, the dysregulation is referred to as chronic inflammation, in which you get continuous activation of macrophages in a tissue with continuous migration of monocytes and other inflammatory cells into that tissue. Again, think about Mr. Clean. If he spreads bleach everywhere, your hands get red trying to clean it up, that's inflammation. Whereas the adaptive immune system, when it gets dysregulated, autoimmune diseases develop. Again, you can think about Marie Kondo getting a little too OCD and piling up lots and lots of stuff and not really trying to decide keeping everything because it's self or throwing everything away because it's not self. So from now on, when I talk about inflammation, and I want to be clear on this, I'm really talking about innate immunity. So the term inflammation is really connected with overactivation of the innate immune system. I think it's a word we throw around a lot because, it's very evocative. It's little fires everywhere. We know what inflammation means. But in this context of the rest of the talk, if I say inflammation, I'm really referring to this overactivation, dysregulation of the innate immune system. So onward to the heart, off the lymph nodes. So I became interested in inflammation in cardiovascular disease a while ago. I would say during my formative years of fellowship, really. And it was based on an evolving hypothesis that innate immune activation was a key feature in heart failure. And this was an early study that showed that the levels of tumor necrosis factor alpha, a cytokine produced by these innate immune cells, was increased in patients with congestive heart failure. This was a study where you had controls. This is chronic renal failure patients as a sort of another control group showing this just not elevated in everyone who's sick, and heart failure patients in which it was much more elevated. These data were more refined and several other studies were done over the years. This is a very good one from what I like to call the turn of the century, in which it was shown that not only was it elevated in heart failure, but the elevation, and this is soluble TNF receptor, so elevations in multiple molecules of this system tracked with the degree of heart failure. So the levels went increasingly up as you went from class 1 to class 4 heart failure. This was a study of 160 patients, all with decreased ejection fraction. And that if you independently looked at the quartiles of levels and survival, independent of VO2 max, there was an increased mortality with increased levels of TNF. So this seemed to be a causative factor in making heart failure worse at the time. And it was hypothesized that if we block this inflammatory pathway, that was overactive and that it would basically be an addition to our repertoire. But unfortunately, the immune system is not like the neurohormone system. So really, we were looking at these elevated immune levels and saying, great, well, there's elevated angiotensin levels in heart failure, and we block that signaling and people got better. And there's elevated catecholamines in heart failure, and we block that signaling and people got better. So if we just block this immune signal, people will get better. And luckily, there had been developed already a drug, Intercept, which is a TNF alpha inhibitor that was FDA approved for rheumatoid arthritis already. And so there was a drug that was waiting and ready for this test. And unfortunately, or fortunately, because we've learned a lot since then, this was a huge failure in some ways, in that it was a negative result. So there were two trials, the Recover trial and the Renaissance trial. One was in the United States, one was international. And as you can see here, this was placebo patients, Q weekly or biweekly dosing of the Intercept. There was no improvement in the number of patients who felt better or got worse. And importantly, when you looked at event-free survival, events being hospitalizations, there was no difference in the two groups. I think a key factor that really stopped this way forward, and in retrospect was probably a good thing, was that Intercept had already been FDA approved for rheumatoid arthritis, and it was so effective for rheumatoid arthritis that they could barely make enough to keep up with the demand. This was before there were multiple other immune modulators for rheumatoid arthritis. And so the company was just like, we're cutting loose on this. We do not see a benefit. There were arguments made at the time that potentially the patient population was too ill and was beyond being helped, and different populations could potentially be helped. But the company was like, we're not giving away the drug free. We're making plenty of money. Our rheumatoid arthritis patients are benefiting. We need it for them. And so that started really, a 20-year, 15 to 20 year, going back to the boards and figuring out, what is the immune system actually doing in heart failure. So most of the early studies after this came out were looking at myocardial infarction. And that's because, of course, the definition of the innate immune system is that it is going to respond to injury. So a number of groups studied in detail how does the innate immune system get activated in the heart, in cardiac injury, and myocardial infarction. And you can see here that one of the major first things that came out of this is that these toll-like receptors, which are the receptors that are activated by TNF alpha, are actually activated by a wide variety of different particles that are released with myocardial infarction or any infarction. So cell necrosis leads to different pieces of the heart, different proteins that then can activate these different toll receptors, which then stimulate innate immune cells. Pieces of extracellular matrix, particularly proteoglycans, can also bind to these toll receptors and activate the innate immune system. And then once you get some tissue injury, you get neutrophils that immigrate, and the neutrophils themselves make products that activate these toll receptors. So then these are all receptors that are on these endogenous monocytes, macrophages within the system that become activated and then amplify this response. It became clear that it wasn't just one monocyte macrophage phenotype that occurred, that actually there were several different kinds of monocytes circulating in the blood that could then emigrate into the heart. And these two types, and this is in mice, where you can differentiate them by a marker LY6, whether they have a lot or a little, they emigrate into the tissue and they have these M1 and M2-like properties that have different contributions to tissue healing. They are stimulated by different cytokines that are released from endogenous macrophages in the tissue. And it's summarized here, where the M1 pathway is mostly concerned with degrading extracellular matrix, eating dead tissue, clearing things away, and then elucidating chemokines and cytokines that call in more of the monocytes, including a second population, the LY6 high, that become these M2 macrophages. And the M2 macrophages quell the inflammation of the M1s and also stimulate pro-fibrotic factors to make matrix and scar in the damaged heart. And these mechanisms are present in multiple different organs injury, but there are tweaks between each organ. We're still not really sure about this back and forth within the heart of changing phenotypes. That's something that needs to be worked on, but a lot of this has been worked out through many mouse studies. And these studies have included ablating macrophage subsets, altering fibroblast activation, increasing the number of stem cells, because one issue with this in the heart is that you don't have stem cells to replace the damaged area. So a lot of different mice have had a lot of myocardial infarctions and a lot of different manipulations of different pieces of the system to give us that understanding that I just simplified right there. And I just want to say our lab has contributed to this. We used a mouse model that had slightly higher UPA expression than normal. We had found that this made these macrophages to be more M2-like. And there had been previous papers with the null version of this mouse that showed that it inhibited macrophage migration into the heart. And this is I could show you about 100 papers like this, where this is the normal infarct in a mouse, where you have this thin collagenous scar that forms. If you increase the M2, you get a thicker scar that is more compact. And actually, these mice had better function in the short term post myocardial infarction. And then if you inhibited the ability of macrophages to come in, which this mouse had insufficient macrophages and proteases coming in, you actually had very little scar formation, and you had these kind of like dead embodied, they call them mummified cardiomyocytes stuck that couldn't be degraded and move on. And below here, this is a stain for activated fibroblasts. That stain cross reacts with blood vessels, smooth muscle cells, which is why you see these, which are blood vessels. But you can also see that when you have more of these M2s, you have more fibroblasts, more scar, less any macrophages coming in, very few fibroblasts, very little scar. So this story, I think the great thing is validated by lots of different mouse models, inhibiting lots of different things. And people are still working on this to this day to see if there are ways that you could modulate scar formation in the heart to make quote unquote better scars and improve myocardial function after myocardial infarction. Now, of course, doing all of this will influence the propensity for heart failure. Obviously, if you have more remodeling of the scar and it's thinner and more stretched out, you have worse function, but it's really an indirect. So I want to move on to what about all the other causes of heart failure? How does the immune system affect that? Because we see plenty of patients we see ischemic patients who haven't had one big heart attack. They just have multiple, it seems. And we have plenty of patients with idiopathic cardiomyopathy, genetic cardiomyopathy, hypertensive cardiomyopathy. We don't see a ton of that here in the Pacific Northwest, but other areas of the country, it's more prominent. I haven't even brought in the whole issue of HFpEF, valvular cardiomyopathy, and now increasingly, as those of you who've been upstairs in the units know, we are now seeing patients with COVID-induced cardiomyopathy that may or may not have had issues prior to getting COVID, but then are coming in several weeks after their COVID infections with rip-roaring heart failure. So heart failure can be thought of as a chronic inflammatory state. And with all these myocardial infarction studies coming out, with the knowledge that there's elevated levels of circulating inflammatory factors like TNF alpha in the body, the model from myocardial infarction got kind of moved to more chronic heart failure. And so again, that there are these circulating inflammatory cells, that they traffic into the heart, and that you have basically a continuous turnover of both of these things going on. So you have M1s making cytokines and degrading ECM, and then you have M2s that are inadequate to block the M1s, but they are stimulating pro-fibrotic factors. And we added to this data also in showing that in patients who were undergoing LVAD implantation, that there was quite a lot of M2 macrophages in their hearts. And so that sort of supported this hypothesis. You'll also see when you see models of this particular PowerPoint object is the most popular for this thing, which is that there's a lot of hand waving, that there's some sort of stress that is continually stimulating this. And really the two big factors that have been overlooked in transposing the acute myocardial infarction healing model to what happens in the chronic heart failure model has been this lots of hand waving. I'm waving my hands in case anybody can't see me, waving my hands, over what this stress is that continues to stimulate these inflammatory factors. People talk about hypertension, and people talk about diabetes and increased redox state. Everyone loves to blame a good oxidative molecule for causing lots of cardiovascular disease. In ischemic, haven't had a big heart attack, people talk about hypoxia, they have chronic hypoxia to zones, and that's what keeps this process activated and continuous. But I'll be honest, it's hand waving. The other thing that has not been taken into consideration is the fact that the heart has resident macrophages in it. So there is a strong resident population. And this has been important because we know that tissue-specific resident macrophages have an important role in other organs, in both both innate immunity and in tissue homeostasis. So, here's an example of favorite ones that people are more familiar with than in the heart for tissue resident macrophages. There's, I think, the Kupffer cell is the one that people are most familiar with. Those are all the macrophages of the liver that are responsible for removing detritus from the portal venous system as it comes into the liver. Any bacteria that work their way from the intestines get eaten by Kupffer cells. We also know that if you have too much fat coming from your intestines, the Kupffer cells eat it, and that can become maladaptive. But those are well known. The alveolar macrophages, of course, we talk a lot about them as being sort of a surveillance program in the lungs to make sure that bacteria don't get through. Osteoclasts in bones are another example of tissue-specific resident macrophages. They stimulate bone turnover so that, like, the same bones we're born with get strengthened over the years until they don't anymore. And then in the brain, the microglia, which are the tissue-specific resident macrophages, have really specific brain functions. They actually, in the turnover of structure, they help with myelin turning over and helping new myelin generate to keep your neurons nice and sparkly fast, and also work on pruning synapses and work on synapse connectivity. So every organ has its own specific resident macrophage with its own specific homeostatic processes that keeps that organ in shape. And similarly, the heart has quite a lot of resident macrophages. And I apologize, this is one of those things where you pull the slide out of the paper and you don't save the paper, and then you have to try and find the paper, which I could never find again. So whoever these lovely people were that published this in 2012, I'll find you and put you here eventually. But you can see, I love this because this is a great marker showing how many macrophages there are in the healthy heart. So this is a mouse heart, and you can see the green is a macrophage-specific stain and promoter. I think this is a promoter, so that only macrophages make it. And you can see not just are they all everywhere, little fires everywhere, but they're very closely intercalcated with the cardiomyocytes. So they clearly perform some sort of role in the heart. And what is that? Well, the thinking is that similar to others, this matrix turnover is an issue. We know from studies where people have been labeled with radioactive substances, that the matrix of the heart turns over in normal hearts probably about every 90 days. So all the collagen between your cardiomyocytes will get degraded and new collagen made every 90 days or so under quote unquote normal conditions. We feel that there's probably some tissue repair that occurs in the heart if there's small amounts of damage. We know these macrophages can come in and they can cloak the cells and prevent the cells from stimulating. If it's just a little bit of a tear, they can fix things up, patch them up. And then we know that cells turn over in every organ, and the heart's no different. The cardiomyocytes themselves don't turn over, as in they are essentially non-proliferative. So if you got rid of them, that would be deleterious. But the fibroblasts within the heart turn over, the many endothelial cells are going to turn over. So there are plenty of cells dying natural deaths that need resident cardiac macrophages to clean them up. Increasingly, though, we are finally learning some of these very specific roles of cardiac macrophages in homeostasis in the heart. And this is a great paper that I recommend everybody go read. And you don't even have to read it. It's in Cell. It was online this fall, and I think it's published. It appears in like the first journal of 2021. Now, I was so excited when I got a paper in JACC, and I got to work with an artist who made the visual abstract for me. And, I've had a couple papers in JACC, and that's super exciting to get your visual abstract. Well, if you publish in Cell, you get an entire movie. So I'm not going to show you the movie. So I recommend you go to it and look at the video. I've just taken some snapshots, but they have an entire movie explaining this paper, which I just find phenomenal. This is the future. So in this paper, they looked at the role of resident macrophages in cleaning up extra mitochondria made by cardiomyocytes. So as we know, cardiomyocytes packed with mitochondria, right? They need it, a lot of energy. But similar, mitochondria age and they get old. And when a mitochondria ages, then there's several things that happen. One is that it can undergo mitophagy, or the cardiomyocyte itself can put it in a little lysosomal structure and degrade it. But a cardiomyocyte doesn't have a lot of time to do that, right? It's focusing on contracting. It's focusing on making new mitochondria. And so although there have been lots of studies on mitophagy and autophagy in cardiomyocytes, this study really illuminates something that had been a question for many people for a while, which is how's the cardiomyocyte spending all this time doing this? And in fact, it turns out that these resident tissue macrophages in the heart are really the ones that take care of most of the recycling of mitochondria. And so here you see this is an exophyr being made, where the old sad mitochondria are packaged, and then they're out into the intercellular space, and then this tissue macrophage here is going to take it up. And essentially what happened is when they knocked out the receptors that these macrophages take these up with, they got a accumulation of these mitochondria outside of the cells and inside the cardiomyocytes. And this led to activation of the immune system, the inflammasome, and poor function of the cardiomyocytes. So they hypothesized that an important role of resident macrophages is that by cleaning everything up, they are tamping down inflammation. They are tamping down these loose mitochondria that would otherwise stimulate an immune system as thinking that they're dead cells, and keeping inflammation at bay. And by doing that, they're allowing cardiomyocytes to expel these and make new energetic ones. So I think this is a really important new direction that the field is going in understanding what a normal resident macrophage is supposed to do. In addition, we know that resident macrophages are sensing the larger environment around them. So this was new data by Corey Levine, and it's not peer reviewed, but I reviewed the paper. I would publish it if it was sent to a journal I was an editor on or I was reviewing. So Corey's group has been looking at these resident macrophages, where they come from, where they go, for quite a long period of time. And he's done his work with Doug Mann, who's the original TNF guy. And so they looked at a model where they knocked out the cardiac, the resident cardiac macrophages and showed myocardial dysfunction. And what they showed in vitro studies here was that when they were stretched, the macrophages actually made growth factors that were favorable to cardiac hypertrophy. So these growth factors were made by macrophages that were stretched, and this could be blocked by blocking TRPV4, which is an ion channel which senses specifically stretch. So here's another potential role for cardiac macrophages. When there's increased stretch in the heart, they make more growth factors for the cardiomyocytes to then compensatory hypertrophy. Again, another interesting paper where when they knocked it out, bad things happened, these particular things. And I really love this paper because we have found that this process of stretch could potentially be part of the mediators in heart failure inflammation. So we looked at changes with unloading. So the opposite, we decreased stretch. Well, we didn't decrease stretch. The patients got LVADs, which decreased the stretch in their hearts. And we looked at, we being Steve Ferris, thank you, Steve, out there, looked at gene expression in cells, in isolated macrophages from patients that were pre-LVAD and then post-LVAD. And looked at expression and saw a very surprising change in phenotypes in which the macrophages had an increase in the inflammatory cells with unloading, inflammatory markers with unloading, and statistically significant decrease in all of these M2 markers. So it seems that with unloading, there was a change in the endogenous macrophage program, which I think in combination with these data from Corey on stretch, and then Matthias Nerendorf's group has also shown a similar reactivity to stretch from cardiac macrophages, goes together with this stretch syndrome induced changes in macrophage function in the heart. And this makes sense when you talk about a myocardial infarction and the healing. We know that the distant areas which are undergoing extreme wall stress become damaged, and this may be part of that mechanism. So really, when we talk about heart failure as a chronic inflammatory state, we now know that these resident macrophages are really what's keeping this in bay, in part, that they are taking care of the damage in the pieces of material that would otherwise stimulate the immune system in the heart. They are keeping the cardiomyocytes happy when there's stress, like increased hypertension in the heart. We don't know about hypoxia yet, but I'm guessing those data are coming soon. And so we know that these perform a protective role, but we also know that they don't last forever. And more data have shown that although they are self-renewing, these cells are with you at birth. They are yolk sac derived that live in the heart. They are self-renewing. There's two forms of them, this CCR2 plus and negative. So at baseline and in a healthy heart, they renew and maintain themselves. However, with time, with aging, with repetitive breaking down of this system where it's just too much for these homeostatic macrophages to deal with, like a hypertrophy in which there's more mitochondria needed and there's more being spit out, and so the macrophages can't keep up with them anymore. These macrophages then die at a greater rate than they can self-renew. And when they die, they are replaced by circulating blood monocytes. And so this over time, we know that these monocytes replace the endogenous and become the resident macrophages, but they have a different phenotype. They are more prone to inflammation. There are many more of this CCR2 pro-inflammatory group than the more maintenance neutral CCR2 negative group. So this is a key factor that we are now understanding and why heart failure may develop over time and why immune activation may increase as you have increasing injury from this stretch and other factors that caused heart failure in the first place. You have increasing replacement of your native happy resident macrophages with blood monocyte derived macrophages. They are more inflammatory, and that can lead to a positive or negative, as you think about it, feedback mechanism. And this is the summary of multiple studies. Again, Matthias Nerendorf's group has been the most prominent in this area in knocking out these populations and showing that it adversely affects healing in response to acute stress. So again, here we go in the chronic inflammatory state, increasing amounts of stress, decreasing numbers of happy resident macrophages, more migration of these M1s and M2s that then are wreaking havoc. So in summary, our current hypotheses are that cardiac macrophage transition from homeostatic resident macrophages to these monocyte ones worsens the chronic inflammatory state in heart failure, that stressors such as wall tension, energy needs, hypertrophy could speed this process along, and that also recurrent release of pro-inflammatory factors systemically could perpetuate this state. So diseases in which we know that diseases in which there's a lot of pro-inflammatory factors wandering around the blood will also accelerate the migration of monocytes into other tissues. And in addition, that specific immune responses to stress may direct this transition between a HFpEF versus a HFrEF phenotype. So the paper that I cited on stretch that Corey Levine had, they actually, and Fareed, you're out there, the TNT mouse. So they took a mouse dilated cardiomyopathy, known sarcomeric mutation, and they knocked out these resident macrophages. And what happened was actually the mice developed HFpEF instead of HFrEF. So they didn't dilate, they didn't thin out, but their stroke volumes were very small. So in modulating this resident macrophage population, you are really, this would suggest modulating cardiac remodeling on a global scale in response to these stresses. So I've talked a lot about mice, and I want to go to humans. So there are, here I'm talking, that was Levine, worse heart failure. There's some supportive epidemiologic data. So one, cardiac macrophage transition worsens. We know with aging, heart failure gets worse. And with time, we're going to continuously have replacement of these monocytes. So that's number one. Number two hypothesis is that recurrent release of pro-inflammatory factors perpetuate and worsen this state. Now, what's interesting is that we know that patients with risk factors for systemic inflammation have worse outcomes. And I think that, diabetes we know is a systemic inflammatory state, and we know that diabetics with heart failure have worse outcomes as one explanation. And we also know that specific immune responses to stress may direct transition from HFpEF and HFrEF. So women with aortic stenosis are more likely to develop HFpEF versus men who are more likely to develop HFrEF. And so I'm going to go over these epidemiologic things and what they could mean. So the pro-inflammatory factors, I think the most interesting angle on this that is very, I think, current is the concept that social determinants of health influence systemic inflammation. So we know now, and there have been numerous studies, this is kind of summed up in this review, that people who undergo an adverse environment, low socioeconomic status, instability, loss, trauma at young ages, and now we would certainly add structural racism and structural sexism to this, that these social pressures actually change brain function in a way that stimulates the adrenergic nervous system. So the sympathetic nervous system is activated. You're undergoing, from these stressors in your life, you undergo kind of a constant flight or fight thing. These elevations in circulating norepinephrine levels through beta adrenergic receptors and the usual have been shown to increase inflammatory potential in these patients. So they have higher levels of NF-kappa B, higher levels of activated innate immune cells in their blood, and adaptive also. And this is, I think, a very interesting potential field of study in the future. That there's actually kind of a cassette of genes that's been defined that go inflammatory genes that are affected, that are reproducible in several different studies like this. And that this increase in systemic inflammation then can increase disease risk, as we talked about. In heart failure, you can imagine that if you have increasing inflammatories of these cells, you're going to have an increasing rate of replacing those happy resident macrophages with these more inflammatory circulating monocytes. And then on the right, it just talks about how this isn't just like a one-way street, but there's different feedback mechanisms. Increasingly, I'll also say that these feedback mechanisms are related to epigenetic changes in the system, but I'm not going to torture you all with epigenetics today. The second big epidemiologic observation we have is that the immune response differs between men and women. And COVID has really nailed this one home for everybody. There are a bajillion papers also on this. That came out in the last 6 months, and that shows that the deaths with confirmed COVID-19 are much higher at every age than in men than women. And I've always found this fascinating because there are clearly way more 90-year-old women out there than 90-year-old men. But you can see this is a nice, this is from Spain, but it's happened in every single country, where for every given age group, men are more sensitive to dying from COVID-19. And why is that? Well, they have different immune systems than women. So it's been well known that autoimmune diseases, those Marie Kondo diseases, are much more prevalent in women, whereas because of data that show that their immune systems in general are more activated at any given time. Whereas men tend to have immune diseases of immune deficiency. So there are certain cancers that are more prominent in men. They're felt to be insufficient immune recognition that they're cancers and killing them off. Also, this was a paper from about 2018, and you can see here that MERS was one of the ones they pointed out at that time that was more infectious in men versus women, MERS being a cousin of the COVID. So it's been known for a while that there are these different biases in the immune system, and I think an area of fertile research is looking at these differences and how can we learn from the immune system and inflammation in the heart by leveraging these differences. So how do you test these hypotheses? Well, mice have a real limitation in studying the immune system because they are not the same as humans. And I was really struck around 2010, a big paper came out that really demonstrated this because they're a common study mechanism of sepsis is cecal puncture in mice. It's gross. Like you just puncture the intestine and let it leak in the inside and look at the immune reaction to that. Well, it turns out when they did more sophisticated genetic array of that immune reaction, very different in mice than humans, in part because the kind of crude theory was, well, mice eat junk and they run around in the junk. And so they have different immune systems for dealing with junk in your gut. This has been overcome to some extent by generation of mice that have humanized immune systems, and also genetic engineering of particular human proteins into mice. So for example, COVID studies were thought to be impossible to do in mice because they don't contain the receptor for COVID, ACE2, but people have genetically added ACE2 to mice and been able to do some effective studies with it. And mice, I think, are always going to be your best model for differentiating very specific pathways of does A go to B, go to C. Human studies are challenged by the fact that you have a lot of genetic variation, and it's difficult to know what a control is. So there are many human studies out there published where they look at normal human versus diseased human. And of course, there's going to be a bajillion differences between those two conditions. So I think in what our studies in LVADs have done and our future studies are that we really need to exploit differences that change phenotype in disease to understand disease in humans, and not look at normal versus diseased, but look at disease at one stage versus disease at another stage. And again, focus when we ask questions less on observation of we found this and more on how can we change factors like we change in a mouse, or take advantage of changing factors to understand mechanisms of disease. So one of these has been taking advantage of some changes. We've been able to induce an inflammation in the nicotinamide riboside study, which I know you've heard Dennis Wang do spectacular presentations on. So I'm just going to remind everybody that one of his findings, and this was published in JCI this fall, that was that in these 4 patients who were given NR, who were waiting for LVAD, so they were very sick, that in circulating monocytes, the gene expression just from giving systemic NR dropped dramatically of these major inflammatory genes. So 4 to 5 days of NR, inflammation factors in the circulating monocytes decreased dramatically. So this is part of a bigger study now to look at quite a few factors, including in giving these patients NR before their LVAD, so that we can get LVAD tissue and look at mitochondrial function as a major endpoint in the study, but also looking at the circulating inflammasome. And importantly for our work, and this is my aim on the grant, is that we can simultaneously look at PBMCs and cardiac macrophages. And now that we know that the NR decreases the PBMC inflammation, we can test, will that have an effect on the cardiac macrophages? Will the cardiac macrophage inflammation also decrease? Of course, we won't know, maybe the cardiac macrophage inflammation decreased and that caused the systemic decrease, but we can see if the two elements track together. And that's going to be key to future studies, because if we can see which elements track together, it makes it more feasible to look at peripheral blood markers as a window into what's going on in the heart. And then the second study that I'm starting, and with Andrew Perry and Zhao, is to look at TAVR patients and to go back to this question of pressure overload and its effect on macrophages in the heart and circulating inflammation and outcomes in LV function. So this is a study to look at patients who are undergoing TAVR, and it's in some ways a recapitulation of our LVAD study. Our LVAD study was flawed in that, the LVAD itself is going to cause all sorts of weird systemic effects that we don't know. So this is no LVAD in place, TAVR day 0, very, nice set time points. We can measure circulating monocytes, and we hypothesize that in patients who have persistent decreases in LV function post-TAVR, that they're going to have persistence of these inflammatory monocytes in their blood, whereas those that improve are going to have a decrease. And the second beauty of this model is that equal numbers of men and women get TAVR, so we can study men versus women. Most of the women that get TAVR are postmenopausal, so then that also helps us with mechanisms. The whole estrogen issue is kind of off the table. And we can just look at genetic issues. So this is the kind of studies that I think are human studies that are necessary to complement the mouse studies and move research forward. So in conclusion, the immune system is fun. The innate immune system plays a critical role in myocardial infarction, and in chronic heart failure, it appears that interactions between resident cardiac immune cells and systemic immunity are critical mediators of the phenotype. We don't have that magic bullet, but will depend on continued research in both mouse and human disease. Thank you.